before you leave. All right, so it's my great pleasure to introduce my dear friend and now colleague, David Albertini. David has taught the egg field a great deal in his years of experience, and he was willing to give our dinner tonight. So David began his world at UMass Amherst. No, UMass.
It started graduate.
Yeah,
Well,
but your undergraduate work was at Harvard.
no, my undergraduate work was at Marquette University.
So never mind. So, so all I know about David really, so you can fill in the background, is that when I came to Boston, David became part of our egg group from which this whole symposium has grown. And at the time, David had gone from Harvard to Tufts, and he was eventually heading up the cell biology, anatomy and cell biology department there. And there were a few different labs in the Boston area that were studying eggs. And graduate students, and as you guys know, who study eggs, you have 10 cells to work with when your companions up the hallway have 100,000 cells to work with. And so the challenges and the database is very different, the knowledge base. And so we would get together once a month. And what really brought us together once a month was the pizza and the beer. Yeah. And we took turns at each institution around Boston. And then at one point in time, we actually, I was mentioning this to somebody earlier, we actually got an influx of funds from a benefactor who wanted us to be able to have a whole day meeting instead of just monthly talks. And so that's how we began this whole day meeting with our colleague, Carol Warner, who is now retired from Northeastern University. And so we began it almost 20 some years ago, and we've had several meetings. And the goal of this meeting is to bring together people, basically people that I need to hear from our work, but that are people who would not normally interact at the meetings they normally go to. So as you know, you tend to go to the meeting of your peers, how many of you ever go to the cell biology meeting? Who here has been to a cell biology meeting in the last 5 years? I'll bet you have, David. Yes. The cell biology meeting is in Boston this December. It is a really wonderful meeting, but you tend to go to your specialty meetings. How many of you have ever been to the circadian rhythm meeting? Have you been to the one down at the beach? Do you just go to meetings? I know. I'll tell you that the cell biology meeting is really the circadian rhythm meeting is really worthwhile. It's at a beautiful resort. Where is it, Eric? It's in
next May. It's going to be in Puerto Rico and Canton.
Oh, that's unusual. Okay. Anyway, it's a really fun meeting. So if you could possibly work going into the circadian rhythm meeting, you will learn a ton, especially about steroid hormone role in circadian rhythms. So anyway, so I want to thank all of you for coming. And it's really my pleasure to introduce David. He's going to talk to us about kind of an overview of human egg research, past, present, and future. And this is particularly important right now because the reason we have Bedford Research Foundation is that you cannot today in this country study human egg activation with federal dollars. There is an amendment to the NIH budget that's passed every year called the Dickey-Wicker. I think it's now called the Dickey Amendment because I think Wicker is not there anymore. And it is specifically prevents federal dollars from being used for human egg activation of any kind. So that's why the foundation was formed, was so that we could do the research that we could not fund with our NIH money. So David's going to talk to us about his perspective, which has been very valuable to us.
Thank you. Yeah.
So for those of you who haven't visited us in Bedford, you should stop by, okay? We were just talking about the team. I had as Ann mentioned, I've known Ann for many years since the Egg Club started. But I had the opportunity in 2018 when we moved back to the Boston area to join Ann and everyone at the Bedford Research Foundation. And it's an intellectually stimulating environment. And the reach out of the SPAR program, the PVSA, et cetera, really keeps the patient in mind amidst our own curiosity about what's going on in reproductive medicine. So today we're going to start with a little quiz. And make sure, I just need to get my pointer working here. Okay, so around the title here are pictures of eggs, not embryos, pictures of eggs that cover the history of some of the highlights in egg research that I'd like to share with you today. Okay, so we'll start with this one. Okay, this is a cross-section of a human morula. Any ideas, any guesses as to when this image might have been taken? Even a decade, pick a decade, like, the 1980s, for example. 1920s, okay, not too bad. This image was taken from the Hertigan Rock collection, the Carnegie collection. The embryo was actually, in quotes, discovered in a woman's reproductive tract in 1932. 1932. This is from the original plate. The actual specimen is located in the Carnegie collection, which is, I think, still in Baltimore, Maryland. Okay. This one, okay, 1938, the first 4-cell human embryo that was really ever seen in the human. Okay, we're going to go through these kinds of studies. This one is relevant because, as Ann mentioned, I worked here at Tufts, and in 1987, I know I'm dating myself, but I really wasn't around in 1932. Okay, just in case you were wondering. Okay. But this particular image came from one of the first very nice fertilized eggs generated by the IVF Center here. Richard Reindaler and Tom Dusabella was actually, and actually we have someone in the audience who was here. Shelley Amster was our andrologist at the time. So that's a little bit of local history. This was just a block away. Any guesses for this? So in 2016, the summer of 2016, 2 very important papers were published, and one was in Nature. This is from the Nature paper, and another one was published in Nature Cell Biology. These papers came from Magda Zernicka-Getz lab. That was the first extended human embryo culture study. This image is actually from the paper of the Ali Brivenlow group at Rockefeller University. And without going into the details, both of these studies were the first to report the incredible self-organization principles that a human blastocyst could have if simply put in a two-dimensional culture. Okay. And finally, this is 6 weeks old. This is taken from one of the recent studies that we'll talk about, where iPSC cells have been used to generate human models of early development. And we'll trace over this kind of pictorial representation a bit of the history of the field of egg research. I really like this. Dan Winkler today was reminding us that those of us from a certain generation don't have access to all the digital versions of our collections of reprints, et cetera, okay? So putting together this talk, I had a chance to dig out crusty old manila folders where I'd been storing things that hopefully someday I'd get to use, but this was a classic paper written by Bunny Austin and Amoroso, two of the founders of egg biology, really in the UK, very much in the UK tradition. And I love their definition here of the egg. The egg is the most unusual cell, not only on account of its great size, but also because it passed through a series of changes here described of so radical a nature that it's commonly referred to as though it were a succession of separate cells. Think about that. Okay, when we think about the continuity of that early developmental process that we're talking about today. Okay, the illustration for this, no, actually, it's about the pictures. They're fantastic pictures of eggs of all kinds, okay, in this article published in 1959. But it sets the stage for both some of the history, the ancient history, as we move to the modern history of human egg research. This is really at the heart of not so much what the research has told us about the human egg, as much as it has about the political, the ethical, and the legal issues that we've confronted over the years when it comes to human egg research. Okay, so we're gonna delve into some of these things. Okay, so we're going to take a quick look at the past, present, and future. I have to admit, unless I'm unconsciously biased about this, much of the original egg history actually took place within a 50-mile radius of where we are right now. Okay, this is Gregory Pincus. Gregory Pincus was an assistant professor at Harvard University before he failed to get tenure and went out to work with Richard Hoagland at a small laboratory in Shrewsbury, Massachusetts, that became known as the Worcester Foundation for Experimental Biology. This institution was a landmark in the history of human reproduction. It's where the original experiments done by Pincus and Rock that led to the formulation of the birth control were started, okay? But it was also because of Pincus, okay, what would launch the very first attempt at in vitro fertilization with human eggs, okay? So what happened? Well, for one thing, MC Chang was one of the early colleagues out there. And as many of you know, whether you're working on mice or hamsters or whatever, MC Chang really set the stage for some of the earliest aspects of egg biology and egg research, discovered the cortical reaction, did the first in vitro fertilization experiments with animals, et cetera. This was all going on at the Worcester Foundation for Experimental Biology. And Bob Edwards is going to come up on multiple occasions, but Bob was the one who was going to build on Pincus's work that eventually led to the birth of Louise Brown in 1978 and the first successful in vitro fertilization case. But there's more, okay? Because if you look back at this, in the 1930s, okay, Pincus and Saunders were amongst the first to figure out that you could take a human egg out of the ovary and that it would on its own spontaneously mature to metaphase of meiosis II. Okay, this was a fundamental discovery in egg biology. And as you're going to see in a minute, this set the stage for the first human IVF experiments. Edwards picked up, as I mentioned, he picked up on the foundational observations of Pincus and started to use human ovaries to obtain oocytes that would undergo maturation in vitro. And in fact, this is one of the landmark studies in the field that we'll come back to later, because it was with this in vitro maturation system using oocytes taken from human ovaries that they were going to go on to the stepping stone to fertilization in vitro. But like I like to say, 50 miles within this radius, things were happening back in Boston because Pincus and now Rock, the clinician, were starting to collaborate. Okay. Pincus invited Rock out to the Worcester Foundation, heard about his studies on animal eggs. Okay. And this launched the very first attempt to do IVF in the human. And my first scientific mentor was a collaborator of Rock's, Arthur T. Herdig. Okay. And Arthur Herdig and Miriam Mencken, who I'm going to come back to in a minute. Miriam was the technician for John Rock that assisted Herdig in the classical egg hunt studies of the 1930s, which formed the basis for our understanding of early human development. Okay. And it's interesting because in the course of this work, I'm not going to go into all the details, but the bottom line is that Rock had patients who were scheduled to undergo hysterectomy for various reasons. These were patients he had seen for many years. All of those patients, there were 68 in total that were staged as far as their cycles go. And they were on a certain number of days prior to having their surgery, they were told to go ahead and have intercourse. And their reproductive tracts, when taken out of the woman, would be used to execute what was known as the egg hunts, the classical egg hunts, from which the Carnegie collection of human embryos was generated. Okay. Okay. And this is the paper. Actually, I have to credit Anne for calling this paper to my attention many moons ago, okay, that was published in Science magazine in 1944, and it declares the first cleavage of a human egg that was fertilized in vitro. Now, those human eggs, if you read through all the footnotes here, were the result of when these operations were done, the ovaries were brought into the pathology lab at the original lying-in hospital. And Herdig and Miriam Mencken would proceed through a very laborious, time-consuming dissection of the whole tract. They were, in the first case, looking to find the early stage embryos and locating implantation sites in the uterus. And from that material, they did their histology, et cetera, and generated what we now know as the collection, the Carnegie collection. But at the same time, the ovaries were used to isolate oocytes from the follicles. And those oocytes were subjected to what I think is probably the first in vitro maturation of human oocytes that in this particular case were used to generate those embryos. Okay. Of course, one of the outstanding questions in this whole story is, where did the sperm come from? And I've never received an adequate answer from anyone. Of course, most of these people are long since deceased. This is Arthur Herdig. This is a photographic rendition of Arthur Herdig. This is John Rock, supposedly peering over one of those specimen trays where the material was being subjected to its dissection. Okay, and from those efforts, which extended from the late 30s into the mid 40s, there were nearly 900 of these human follicular eggs that had been isolated and studied in the course of this investigation. And of those, we think one was clearly fertilized as far as they could document it with the primitive microscopes, as I think you can see here, that they had available to them at the time. So it was this in vitro maturation experiment with those eggs back in that time span that actually led to the first IVF. This is a rendition of Miriam Mencken. And the efforts of Mencken and Herdig and Rock resulted in this embryo. This is from the original science paper. The tools were cheap. The media was simple. It was basically a salt solution that had serum from the patient in it, and the hopes were high. But there was never much press made about this. Okay, again, you have to think 1944, was society even ready to hear that people were doing this kind of thing, okay, under those conditions, all right? So how did this play into what we know today? We're working towards where we're at with human egg research today, which is a pretty troubled state when you look beyond our myopic eyes as scientists and research investigators. When you begin to look a little more broadly at the ethical implications, at the social implications, you can begin to ask, why are things like the Supreme Court decision last June, okay, why are these things happening? Okay, a lot of this has had to do with how the original work on the human egg was represented to not just the popular press, but to people in general. And this is a book that that was drawn to my attention, I think about 10 years ago now. It's written by Lynn Morgan, who's a professor at Mount Holyoke University. She's actually a social anthropologist. And she tells the story in this book, Icons of Life, A Cultural History of Human Embryos, of how one day she ventured into an old building on campus and found these glass containers that had all sorts of stages of human fetuses, et cetera, in them. And that piqued her interest in trying to understand more, not only about where did this material come from. It was obviously being used as teaching material at the time, okay? But how did this get released into the public eye, okay? And what were the consequences of sharing human development and this kind of material with the public, okay? So it's a very interesting read. Again, sorry about that. The chronology here as part of the history, again, goes from the Hertigan Rock egg hunts, okay? And interestingly, all of those studies, there were a series of, I believe the Carnegie series ended up being 32 stages of early human development, starting, by the way, with the 2-cell stage. They found 1 2-cell embryo, okay? 4-cell, et cetera, all the way up to, I think, week 14, they had material. All of that material was never published in what we would call the popular press. It was published in what are called the Carnegie Contributions to Embryology, which is basically hidden from the public for over 25 years before that material was finally adopted as textbook material for many of the human embryology books that appeared at that time, both in the UK and in the United States. But it wasn't out in the public's eye, is the point to keep in mind. Okay. We mentioned the first IVF experiment, again, with exception to this one paper, not much was heard. Bob Edwards came on the scene in the 60s. Interestingly, it was the same kind of in vitro maturation experiment that led Edwards and Bavister to attempt the first in vitro fertilization over in the UK. And of course, the result of that, and as we say, the rest is history, was the launching of contemporary assisted reproductive technologies. And our dear friend and colleague of the past, Dr. John Biggers, who used to be a very active member of the Egg Club and of all of the previous meetings of this sort, chronicled the history of many of these early events. If anyone's interested in pursuing it, he actually was really one of the scholars, I would say, not only in terms of the biology of the egg, which he did so much work on, but also the historical context. And it was from that book of Lynn Morgan's that I took this quote just to give us some pause to reflect on where things were at when these specimens finally were discovered and brought to the attention of at least the medical community. And as she states here, the Carnegie Human Embryo Collection is a link between the past, when embryo specimens were prized scientific objects, and the present, when encounters with dead fetuses are regarded as distasteful and macabre. This decoupling of the specimens from representations of them was a significant turning point in American culture. Afterward, it became harder to talk openly about pregnancy loss or to admit that specimens afforded scientists a better understanding of embryological development. So there was some darkness cast on this back at that time. Now we're talking 50s and 60s, which I think is going to play out. I'm going to just skip this. Okay. That's some history. Where are we at now? Okay. Again, we kind of left off that Edwards and Steptoe produced the first successful IVF cycle. Okay. This is a field that has grown tremendously since 1978. And with it has come, with it has come some interesting trends. I'm going to come back to this paper in a minute, but if you look at the gray shaded portion of this graph, this is tabulating the number of publications in reproductive medicine over the time span. I believe it was 1999 to 2019. Okay. This is a significant proliferation of research papers. Okay. Egg freezing is a huge portion of what we presently experience in the world of ARTs and egg research. Okay, we're going to say a few words about some of the political and social issues that surrounded not only the introduction of egg freezing, but its application. It's now widespread application in our culture. Okay, and let's just start to do that. So, thank you, Tiziana. You introduced us today in your presentation to the powerful influence that the church had at that time. I believe that dictum was the one that you cited, was dated in 2004, okay? But there were restrictions imposed by Italian law number 40, okay, on the use of human eggs in clinical practice, okay? From my take of this, I did not know your twist on it, but your research actually benefited from the fact that only 3 eggs from a given retrieval could have been used for clinical purposes, all right? What it did more than anything else was it restricted the fertilization, it restricted the cryopreservation of human embryos. This was a Vatican decree, okay? And I took some of this from the late Dr. Flamini, okay, who was very much involved as a representative of the medical community with communications with the Vatican. But the Catholic perspective in human reproduction is based on the concept that the human being is to be respected and treated as a person from the moment of conception. From the moment of conception. In this paper, the ethical implications derived from such a principle are discussed, okay? This was really the start of imposition, political imposition of restrictions on the use of human eggs, Okay, to my knowledge, I think Italy was the first to step up and impose these kind of restrictions. And this story, okay, was something that was published, actually, Stephen Hall published this paper. It wasn't a paper, it was an article that he wrote after spending a week in my laboratory here at Tufts. This was back in the 1990s, and in John Van Blerko's lab, coming to realize that maybe this definition of life starting at conception should better be viewed as part of a continuum, where for those of us who are eggheads, okay, we think that it starts when the egg starts to develop, okay? So that this would be viewed more as a developmental continuum. But nevertheless, this stood as a very significant step in the political intervention of work that was going on. Now, keep this in mind, because today in ARTs, generating embryos is the name of the game. Okay, so doctors like David Keefe, see their patients. They want to gain as many possible eggs as possible from those patients, see that they're fertilized, and in an ideal case, doing PGTA, etc., the patient gets some number of embryos, euploid embryos, in an ideal world that are stored away that can be used for single embryo transfer at some point. Okay. It's hard to overlook the fact, and you mentioned this in one of your slides today, David, that even with the euploid embryos, the failure rate is still very high. It's still approaching 50%. So it's not enough to know that our genetic testing is necessary and sufficient to identify a subset of embryos that have better chances of producing a baby, because there are more factors than simply the genetic ones that come into play. And I think there's a better awareness of that in the egg research field now. So in some sense, we have to deal with this basic biological fact that pregnancy failure is common in our species, if you will. And one of the questions that we'll come back to is, could a deeper understanding of the mechanisms underlying human embryo development improve ART success rates and diminish pregnancy loss? And that, you'll see, we'll close with when we talk about some of the new work on embryo models, okay, that are being produced by stem cells, etc. Okay, so just again, in terms of where we stand at present, okay, these are some of the trends, publishing trends, that I think bear witness to the fact that things have changed over the years. So for example, remember, Louise Brown was born in 1978. Okay, so this was really the first decade of human ARTs where getting eggs was still a challenge, as was stimulating the ovaries. So that decade of research really, this was a field searching for an identity, I think, in a broader societal context. But it was dominated by pharma. More, better drugs to do ovarian stimulation was kind of the driving force. You didn't see a lot of work on embryo culture media. A lot of the laboratory techniques that was to come. Okay, in the 90s, there were still just a handful of journals dealing with the topic of reproductive medicine. But I think this extends very much to the topic of egg research for today. Interestingly, not only were there few journals to publish your work in, but this was at a time when the field was also still dominated by academic institutions. These were card-carrying OBGYN departments that prized research endeavors, et cetera, and wanted to really deploy, I think, principles of real scholarship and research on this foundational work that was going to be done. 2000, we started to see what I like to refer to as more of the boilerplate treatment years, varying a little bit of this hormone or that, timing, et cetera. As again, cases were certainly growing, and study designs were getting a little more complicated in terms of what we would have called the relevant egg research of the time. And of course, in this particular decade is when the claims for evidence-based medicine became a driving force in this area. And of these things, how to select the best sperm, how to select the best oocytes, how to select the best embryos, Denny will attest to this, really became a prevailing force in the field, all right? With again, the thought that we, mortal embryologists, scientists, physicians, would have some magical insight into which of these embryos was going to make a baby, even though no matter what we did, half of them would fail. Okay, so just keep that in mind. And then I think really what we've seen since 2020, I think the embryology laboratory has actually assumed a prominent position in patient management, in treatment, et cetera, but it's been a long time coming in many ways. So growth, as I mentioned, has been pretty remarkable. Okay, this just happens to compare, cycles, so-called cycle success rate. How you measure that over these decades went from how many fertilized eggs you had, how many cleaved, how many reached the blastocyst stage, how many established a chemical pregnancy. But fortunately, by today's standards, I think the field is moving more and more to ask the question of the eggs we collect, how many actually make a baby? And you can thank Denny Sakis and Pasquale Patrizio for first asking this question. I think your first paper was in 2010-ish or 9. Where they made the effort to say, of these eggs that we collect, how many will actually result in a term pregnancy? And correct me if I'm wrong, but I think your original answers were somewhere around 10% or less. Okay. And your more recent results are in the same ballpark, or?
They were less, and then it's about 10, 11% now.
Okay. With what?
In the better patients.
In the better patients, okay? So I think when you talk about human egg research, to me, this is the kind of result that says we don't know enough. We don't know enough. We need more fundamental research. I think striving for better ways to understand not just activation stimuli, but I think what we've heard today is a really telling point. We've kind of looked at the sperm as donating one thing. Give me a genome and I'm out of here, okay? And we've been fascinated by the sperm centrosome, okay, for a while. Even that, I think we're starting to shape our thinking a little differently, maybe a little more open-minded about exactly what the sperm contributes. But look at what we've been able to do today. I think we put these sperm factors, the RNAs, the exosomes, et cetera, on the map as something really worthwhile to pursue. And freezing, as I mentioned, has been at the heart of this. So this is that one graph. This just came out in JARD. And actually, it's an interesting group. They're more or less epidemiologists. Bernard Revitsky just became the new director of the Hastings Center in New York City, which is a well-known bioethical institute. And they analyze this data not simply as a function of the publications that have been going on in this area, but they broke it down into a variety of topical areas, okay? And their conclusion essentially was that we call on the research community to promote more international collaborations that are focused less on explored reasons and divert more attention to questions of cost, access, knowledge, and attitudes, which is, to me, sending the message that we need to look at the whole patient. This isn't about the science. I think, as Dan Winkler was suggesting earlier today, looking at the ethical, when you think you know what the ethical implications are, most likely we're well short of really thinking deeply enough about that. So what about the future? We have an interesting world in egg research now. Okay. We have heard lots of talk about making gametes from stem cells. Okay. And really the only strong evidence for this comes from the Japanese labs that have turned ES cells and iPSC cells into sperm that work and oocytes that work. They have been able to recapitulate gametogenesis in mice by carefully designing and implementing experiments that mimic nature with respect to the development of gametes. Okay. Certainly gene editing and CRISPRs very much on the map now. Get these eggs, okay, and begin to implement these gene editing tools. What are the consequences going to be in the short term in terms of actually addressing some sort of genetic defect, et cetera? Or, is there going to be some real therapeutic value down the road? We heard today a bit about the mitochondrial replacement therapy from Bjorn. I think we're starting to see, we're at the dawning of seeing some of this fundamental egg research work published, not so much in the specialty journals where it has normally been published, but going to higher profile journals. Okay. Denny is part of the group with Carlos Simon that just did this lovely piece of work that was published in Cell, okay, exploring, among other things, other impacts of stress due to trophectoderm biopsy on DNA stability, et cetera. And I think, we're probably at just the dawning of some of this fundamental research, egg research, I would call it, appearing in the CNS journals, okay, very high profile journals, some of which are for profit, like Cell and Nature, where we haven't seen any of these papers published yet, are not for profit, whatever that may mean. I'm just going to close with a couple of this paper. I think was a very high profile paper. It was marketed very strongly by Nature and many other high profile magazines. And then we'll close with just a quick discussion of the so-called synthetic embryos. So very nice work. For those of you who were at the ASRM meeting a year ago, Nicholas Plachta presented the results of this collaboration with the Carlos Simon Foundation and Boston IVF, really beginning to tease apart some of the cell dynamics in the human embryo. And actually, there's a fair amount of mouse embryo data in here as well to kind of compare and contrast. Kind of paper that you expect to see appearing at higher with sort of higher impact journals. Okay, I think that among the other things that are suggested in this work are, interestingly, that the biopsy itself, the trophectoderm biopsy itself, may be actually inducing a level of DNA damage due to shear stress that had not been previously anticipated before. We were just speaking very positively about Richard Scott, okay, who has been a huge proponent of trophectoderm biopsy not having any effects. Okay, so this I think is important egg research, not only because it's telling us something about the blastocyst at that stage, it's also informing us about the technologies that we have adopted and the ones that we need to look to in the future. Okay, and as we mentioned before, given that ART embryos are showing aneuploidy rates as high as 90%, The question is, could models of human development provide a platform for understanding pregnancy loss? And that's what I'm going to close with, which is not only the fact that we've come to realize that pregnancy loss is a multifactorial phenomena well beyond the genetics of the conceptus itself. But we're now at the stage where people are using these models generated from embryonic, actually mostly from iPSC cells, to generate not only looks like it's gastrulating, smells like it's gastrulating, tastes like it's gastrulating, okay? And again, high profile journals. And are these models, are these studies really telling us things fundamental that could be applied to our understanding of early of human egg development? That's a question that is yet to be fully addressed, okay? These are just two of the recent studies. Interestingly, this one from Jun Wu's lab also it gets into the whole business of, most of the earlier studies in human eggs trace derivatives of the trophectoderm and the inner cell mass in extended culture of actual human blastocysts, okay? This particular study now is venturing into the area of the extra embryonic membranes, the yolk sac, okay, the primitive endoderm, et cetera. These are very different constituents that also mediate very important aspects of implantation and sustaining development. So could these could those be informative in terms of understanding some of these complex developmental processes? So these so-called human embryo stem cell models, as I mentioned, they are recapitulating using ESCs or iPSCs development from day 5 up to day 13 stages. The models have now been extended beyond the day 14 turning point, which we can discuss if you like. Publications of pressworthiness are now appearing in journals outside the traditional purview of reproductive medicine, presenting new technical and legal challenges. I would harken back at this point and ask us, how the word gets out there, what the language is. And you were encouraging me very much to think about going in the direction of the linguistics, et cetera. And I think that's a critical matter moving forward with this kind of research. We'll see if we can put it into the right context. Okay, so the language needs to be addressed to all the stakeholders, not just the scientists, not just the patients, okay, the physicians, okay. We need a language, we need a way to speak to all stakeholders in the business of what we do with these eggs we collect, okay. Will history and the research of human eggs inform us in the future? We don't know yet, okay. Meaning is changing over time. Look at the world we live in today, okay. In a way, being put on the back burner relative to the wars that we're trying to get through, relative to the political corruption and disruption that we have in this country. Yeah, maybe sitting in the background for a little while is a good thing, but who knows how that will play out. And of course, I think, probably one of the most devastating consequences of this has been the move in this country that led to the Dodd decision. So I personally want to thank all the speakers today. I think the program was really rich. I think everybody delivered. We learned a lot of new things, okay? And hope that you've all enjoyed the meeting and that we'll see you back again next time we do this. Thank you.
Thank you, David. That was perfect. Does anybody have any questions for David? He's really good at answering questions, but they're not always short answers.
I do have a question.
Ann knows me well.
Jose,
That was great history for all of us. I mean, but I think that
Want to use the microphone?
We don't realize how much damage the Dickert repair amendment have done in embryology research, because we left research in the hands of industry. And everything that's been going on for the last 20+ years, either sponsored by IVF clinics that are well endowed, or by pharmaceutical industry that have their own interests controlled. So I have a lot of. I actually admire the work by Hannah and Gabe on the synthetic embryos, but nothing justified for the real thing. I think we cannot study this unless we really have the real thing in hand. What are you going to model based on what? Mouse, based on the cow?
Yeah.
We don't have any knowledge of what really happens to the human embryo beyond that. So I hope that one day we can just have a real discussion about how to change funding for embryology in the US. I know Europe is moving very cautiously. Asian countries are a little bit more aggressive, but we always take that with a grain of salt in terms of IRBs, etc. So industry's been playing a role in IVF. I didn't know this until I started teaching a class for my students. The Catholic Church started the whole Serrano. Catholic
Yeah,
Church was the
That's
one
right.
Yes, the nuns.
Yeah,
provided,
sure.
The nuns.
provided
Yeah.
the urine to isolate
Yes.
Yeah, the nuns.
FSH for the first time. So Serrano grew up thanks to that.
The nuns, the menopausal nuns.
Right. HMG.
So God bless the Catholic Church. Anyway, I just think
It
that
was the nuns, right?
Yeah,
That
yeah.
collected the urine from the
Yeah.
convents, right?
Menopausal nuns.
well, I just
Yes,
think that
that's right.
until we have a real funding with no biases, and I appreciate Giampiero telling us about the follow-up on the kids from different types of IVF procedures. But you guys are still private or funded by foundations. I don't know, Giampiero tremendously. But I think that these people should be encouraged to continue their research, but this should be a way of having funds that don't have to be constrained by private.
Yeah, you're touching on a hot spot, Shelley, for sure.
And the money that's being made from the patient needs to come back to the research so that we can continuously improve. And I don't know if that trend is, if there's a movement towards that. That's what bothers me, is the amount of money being made at the patient level and not going back into the research. Maybe Denny can talk about it because he's on the other side. Can you see that money? coming
Shelley,
through?
I can tell you from experience that when you make that point to big medical meetings where there's a lot of IVF programs, you don't get invited back.
No, I know. That's why I'm saying it in this room. Because it bothers me.
I mean, 1% tie from assisted reproduction to basic research would go very far. 1% tie.
I know.
A trend that was obvious, I think, at the ASRM meeting this year. They have these exhibit halls that surround and et cetera.
Small groups.
No, their boots are getting bigger. The
Oh.
show is now get to sponsor their own little special seminars and things, okay? Which, as you might think, are talks that are biased towards using their product. And that's why they do it. And organizations like the ASRM benefit greatly from this. In fact, I was talking to Dan Carey, who's their chief financial officer, and he told me that the annual meeting generates, he said, 43% of their annual income. It's the major source of revenue. But I
And
think
Serrano
we're
is
just
only a very small part of infertility anymore.
Absolutely, and the programs that they are sponsoring, which is not to belittle them, are extending more to alternative family structures, social programs, et cetera, which is a good thing. But your question was, are they putting any more into fundamental research? And I do not see that happening.
I want to let everybody go, but I want to just make one comment to Jose's part. So before the Dickey Wicker Amendment was put in place, the NIH, this was during the Reagan administration, the NIH organized a study section for study of human reproduction. And it was in, I don't know, Reproduction Center. You may remember this too, David. And so they had a study section that was going to study, you could propose a proposal to study early human reproduction, just like they were doing in England. The caveat was that they needed that proposal to be reviewed by a special ethics advisory board to the NIH on human reproduction. And it was to parallel the board that was put in place in England there, that's what was going on in England at the time. So to keep any of those grants from being funded, they could be, so there was a very famous protein chemist called Oliver Lowry. If any of you've ever done a Lowry protein, right? Oliver Lowry's daughter was infertile and she had been through a few IVF cycles. And he was incensed at the lack of research behind what his daughter was going through. So he wrote a new grant proposal every year. It was reviewed by NIH study section. It was granted a very high score, but they solved this by never convening the ethics advisory board. So there was never an ethics advisory board that could review the grants on human reproduction so they could be funded. And so finally people just stopped submitting them. So this was like a government trick. Then when that caveat went away, and I can't remember who was getting rid of the ethics advisory board, is when the Dickey Wicker Amendment got put into place. So the biggest problem with all this is not that we don't have federal funding, it's that and this is the problem with the Roe versus Wade, we've never really had a good public discussion of how to do this, right? There's no public discussion of we need to do this and we need to do it under proper oversight. And we need to get it going, whoever funds it. And so you're right, it's funded by pockets of people. It's funded by us that, set up a private foundation so we could do it. But the biggest problem is that there's no public push behind this to make this better and so that we can really understand it. I think that's the bottom line. So hopefully everybody will go. Is that right, David? I mean, you remember that, right?
Yes.
And so I think everybody could go home tonight and we can try to figure out, we're doing what we can, but until there's a real public discussion of all aspects of human reproduction and human control of reproduction, it's going to continue in these pockets of piecemeal with people who have a lot of at stake are doing the funding. So thanks for bringing that up, Jose.
Well, I was just gonna say, for Shelley's point, what's happening now is that this growing presence, private equity. In New York City alone, there are 12 IVF centers backed by Wall Street. They incentivize the doctors on what they call a conversion rate.
Yeah.
The number of people that walk in the door, and the percentage of that end up with an IVF retrieval, either egg freezing or, which, okay, fine. But what's happening is they have so penetrated, traditionally IVF was sort of the cool, the lady golden egg for OBGYN departments. And in my own department, they traditionally funded everything, all the, we had a pediatric age clinic, somebody who would go there, nobody had insurance. That's okay, because we subsidized it. Now that, we are associated with one of the networks, we're still NYU. So what's happening, it's so penetrated our field, and now a lot of people on the board that certify programs to train are advocating we reduce research in the fellowship.
Right.
This was traditional,
Right.
as part of your formation as a reproductive endocrinologist was to have even a taste. God forbid you might actually get interested in it, you're truly included. I went into it just kind of interested, but now I'm committed.
Yeah.
But now
It's a vacuum that got filled.
we don't need that anymore. They said we need bodies out there because we have a shortage with so many people want to freeze their eggs. It's driving up the recruitment costs. So they're advocating strongly that we shorten the fellowship by a year and that we get rid of all research during the little training so that people can get out there and help us make money. and treat patients.
No, we understand. I'm serious that I don't get invited to any of those meetings anymore because I would always start every talk at those meetings when that are put on by drug companies and you're at this really nice hotel and you're gonna talk half the day and play on the beach or ski the other half.
Like doing less of it now, because of the Sunshine Act, so they're very difficult, like, you know, on beat.
Yes, I know it's
Fucking
different now, but
companies
even
do.
so, you speak to that group of people and you say, you guys, if you would tithe 1% of your income every year, you would know which embryo to put back.
But the first,
We would have done the basic research to figure that out.
the first time I went to one of these meetings, I had spent 3 years with Edith Schwartz at the program at Northwestern doing translational reproductive research. And then OB/GYN residency, my first meeting, I get on the elevator and a drug rep got on and said to the other, have you ever been to this meeting before? The other drug rep said, no. He says, unbelievable. It's the only place in the world where you can watch people that make $500,000 a year stand in line for 45 minutes to get a free pen.
Wonderful.
And I go down, and you know, there's like, I don't know, James Watson or somebody giving the keys, and it's empty. And then there's this huge exhibition hall with everybody with little Cupid dolls and pens with worm and magnets. It's unbelievable.
And you would have thought the things would have changed when they didn't have to pay us to go to their fancy caviar dinners and everything. And have lunch and learns, but it didn't change.
Yeah. Thank you, everybody, for coming. Everybody, please take down your posters. We've learned
That there is a poster board mafia in Boston. So if you want to make if you want to go into an interesting business, there's only one company in Boston that provides poster boards. They charge a fortune and they're about to come and take them.
Monopoly.
It's a monopoly.
Close.